Hello there, and welcome to this Khan Academy tutorial on AS level biology. In this video, we will look at part 2 of the immunity topic, covering antibodies and vaccinations. So let's get started. So here are the aims of this tutorial. We will describe the function and the structure of an antibody, and look at what monoclonal antibodies are, explain the difference between active and passive immunity, understand the importance of the vaccine and the issues surrounding it, and also finally look at antigenic variation. So let's look at the structure of antibodies. Antibodies are soluble proteins made by B cells. They are made of light and heavy chains that are joined together by disulfide bonds. There are many types of antibodies, but all have the same constant region, as shown by this green oval. This means that this region is made of the same genes. However, different types of antibodies have different variable regions, with specific shaped antigen binding sites, making them complementary to one antigen only. Although you don't need to know this, it is useful to understand that this occurs because the mRNA region correlating to the variable region of the antibody is spliced at different areas. A point to note is that all monoclonal antibodies have the same variable region, hence why they're named monoclonal. Now let's move on to how these antibodies function. Complementary shaped antigens bind to the antibody, forming an antigen-antibody complex, a very similar theme to an enzyme-substrate complex. However, since antibodies have two antigen binding sites, as shown in this diagram here, they can form two of these complexes, unlike enzymes which generally form one. The advantage to this is that this allows agglutination, also known as clumping of the pathogen. The clumping of the pathogen means that the pathogen can no longer infect cells or release toxins. They are essentially neutralized. These agglutinated antigen-antibody complexes are engulfed by phagocytes by the process of phagocytosis. Now, I've shown previously that monoclonal antibodies are antibodies that have the same size, shape, and variable region. These antibodies are made by plasma B cells. Due to their identical nature, the antibodies are specific to one antigen only. Monoclonal antibodies can be made artificially via the formation of a hybridoma, which you may remember from GCSE. I'm not going to go into too much detail here, but essentially a plasma B cell is fused with a tumor cell, which in this case is shown as myeloma cells in the diagram, to make identical copies of the antibody. These monoclonal antibodies can be used to provide passive immunity or immunotherapy in cancer. This is because cancer cells have foreign antigens, and immunotherapy would provide targeted therapy. Another use of these monoclonal antibodies is in the ELISA test, which we'll see in a later video. So there are two ways in which immunity to a pathogen can be achieved. One is active immunity, which involves production of own antibodies after exposure to the pathogen. And this can be done in two ways. You could catch the pathogen naturally off someone, or be given artificially in the form of a vaccine. Passive immunity, meanwhile, involves being given antibodies from a donor. So this means that the individual doesn't make their own antibodies, meaning that they are not actually immune to the pathogen. Again, passive immunity can be achieved in two ways. Naturally, newborns receive antibodies from the mother's breast milk, hence why it is better than giving powdered formulations. On the other end, like we saw in the previous slide, people can be given artificially made monoclonal antibodies. So here is a summary of the points I discussed regarding passive and active immunity. Remember, in active immunity, memory cells are made, hence why the individual becomes immune to the pathogen, while in passive immunity, this doesn't occur. In active immunity, immunity is lifelong, while it is short-term in passive immunity. Generally, vaccines are suitable for healthy individuals, and passive immunity are suitable for immunosuppressed individuals, as their weakened immune system cannot handle the pathogen. Now let's move on to vaccines. The word vaccine comes from the Latin for cow, and why this is because of its history. Edward Jenner, a man not related to this woman, in case you were wondering, was a famous immunologist and doctor who injected cowpox to individuals to reduce the severe symptoms that occurs with the deadly smallpox virus. As seen in this image here, this is the first patient who received the life-changing treatment, 8-year-old James Phipps. Though this may be unethical today, let's understand why this worked. So, cowpox is a virus that had a similar shape to the smallpox virus, and this meant that the memory cells made for the cowpox virus could identify the smallpox and kill it before it even caused symptoms, especially severe symptoms. Nowadays, vaccines contain antigens of the pathogen or attenuated, also known as weakened, pathogens. Let's understand why vaccines are important. Vaccines generate manageable immune responses, causing memory cells formation. Vaccines are often injected rather than given orally, as oral administration of vaccines can be digested by stomach enzymes. You might have come across the word booster vaccine, and why it's needed can be explained by this diagram. The initial vaccination is similar to a primary immune response, so a small amount of antibodies are made. The booster vaccine contains the antigens of the pathogen, and this results in further exposure to that pathogen, and hence more memory cells can be made, resulting in a much faster immune response if the person is to contract the virus or the bacteria in future. So giving a booster vaccine is similar to having a secondary immune response. The benefits of vaccines is that if a large population is vaccinated to a pathogen, they become immune to it. This reduces the spread of the disease as fewer individuals will carry the pathogen. There are, of course, some issues surrounding vaccines, just like any other drug. Let's first look at the ethical issues. As vaccines are initially tested on animals, people worry about how well these animals are looked after. Moreover, some people dislike the idea of animal-based vaccines. Despite the benefits of vaccine, there are many reasons why fewer people may take up vaccines. First and most importantly are the side effects that are associated with them. The side effects of vaccines are generally minor, such as having a small fever. However, rarely they can cause anaphylactic reactions, but again, this is very rare. Additionally, some vaccines are expensive to make, so people and even countries may not be able to afford them, and hence people cannot be vaccinated. Also, insufficient amount of vaccines may be available because of the low production rate. So in the final part of this tutorial, let's look at antigenic variation. Mutations, which is defined as a change in the DNA base sequence, can change the shape of the antigen of the pathogen. This is called a pathogenic variant. You may be familiar with the Delta and the Omicron variant of COVID. However, if the mutations cause significant changes to the antigen of pathogen, we get a new strain of the pathogen. Memory cells cannot identify new strains of the pathogen, while they may identify a variant. So if a new strain comes along, then the body's immune system starts again from the primary immune response. Well done for sticking to the end of this tutorial. Here's a recap of the points we covered. As usual, use them as questions to consolidate your learning. I thank you for watching, and I will see you in the next one.